7, I would like to call upon Mrs. Thelma to give an introduction about the speaker, Professor B.K. Thelma.
Good morning, distinguished members of the panel, respected staff and fellow students. Introducing our guest of honor and speaker is a great privilege. I feel elated to introduce our next speaker, Professor B.K. Thelma. Dr. B.K. Thelma is the Professor of Department of Genetics, University of Delhi. She has completed her Master's in Zoology from Biotechnology University, her Ph.D. from University of Delhi, and her postdoctoral fellowship at Human Genetics Lab, Switzerland, with Professor Thelma. She is the principal investigator and coordinator of the Center of Excellence on Genome Sciences and Precision Medicine funded by the Government of India. She is also the coordinator of a gated project on newborn screening for inborn health of metabology of intelligence. She started her career as a lecturer at Department of Genetics, University of Delhi. She has also been visiting scientists at prestigious institutions abroad and undertook several international research collaborations. She established the DNA-based diagnosis facilities for President X syndrome with financial support from Department of Biotechnology. She has also served in many expert committees of Academies, DVD, and CAR. She was the CSIR core officer and became associate in Department of Zoology, University of Delhi. Her main area of research is history genomics, and her projects include genetic analysis of complex traits, including brain and integrative disorders, and many forms of neurological and neuroscientific diseases. Her most recent engagement is in polygenomics, wherein individuals can be deep phenotypes based on principles of Ayurveda. She is the recipient of various national and international awards. She is the fellow of Indian National Science Academy, Delhi, Indian Academy of Sciences, Bangalore, National Academy of Sciences, Allahabad, and Lacy Post Fellow, Department of Science and Technology, India. She was the Vice President of Indian Society of Cell Biology. She is a member of International Commission on Clinical Use of Human Germ-like Genomics University, Scientific Advisory Committee to Prime Minister Government of India, and assigned the HN Research Group. She has been distinguished finalist at the Max Planck Institute for Molecular Genetics, Germany, Department of Psychiatry, Israel, Department of Psychiatry, Western Psychiatric Institute in the University of Pittsburgh, USA. Currently, she has been heading several research projects and successfully completed more than 25 research projects funded by national funding agencies. She has guided 30 Ph.D. scholars. She is serving as a reviewer for several peer-reviewed journals in the field of human genomics and molecular biology. She has published 130 papers in reviewed journals to her credit. She has received the extramural research funding of around 50 crores. We are immensely pleased to have you here with us today for this prestigious lecture workshop. I thank the head of the department for giving this opportunity. Thank you.
Thank you, ma'am. Professor B.K. Thelma to present the third session on recombinant DNA technology and gene therapy.
Very good morning to all of you. I hope you can hear me well. Okay, all right. Okay, I will already start sharing the screen with you. But before I do that, I just want to tell you that while the topic said recombinant DNA technology and gene therapy, I was told by Malati that, there are undergraduate students and it's a mixture of everybody. So I decided to kind of, use my discretion to kind of build from basic genetics, human genetics, and then take you through till the end to talk about gene therapy. Of course, in the limited time, we cannot talk about technological details too much, but I will give you an overall idea so that you are able to appreciate the area as a whole. So if I may now start sharing the screen with you. You'll have to tell me when you can see it well. I'll see entire screen, I think is better.
Okay, ma'am.
Yeah, okay. Login. Yes. Check your name. Can you see?
Yes, ma'am. We could see the screen, ma'am.
You can see the screen? All right, but I can't see it now. Let's see. I can't see my own screen, so maybe I should just stop and do it on here. Okay, I'm just going to stop for a minute and I'll just do a browser share.
Okay.
Only a window I'll do, okay? So, is this good?
Yes, ma'am.
Okay, perfect. All right, so I changed the title a little bit. And it's human genome analysis and leads for genomic medicine. So when I say that, it's genomic medicine can encompass a variety of things, but I'll try and explain to you what exactly I mean. To start with, I also have to tell you that my talk is largely restricted to human genetics, medical genomics, and associated translation efforts. What does this mean to you? I wish I was there in person so I could have interacted, but let's hear you say something. What does it mean to you? What do you see in this picture? Quick. Okay, you see a whole lot of phenotypic variations. It doesn't matter which species or humans you talk about, you have a lot of phenotypic variations. Okay, underlying these phenotypic variations are the genetic variations. All right, now you can have this as I'm showing you in this picture in plants, microbes, all kinds of organisms. The variations will be there, but our focus is going to be what are the variations which are going to make a difference for health and disease. When do you talk about a therapy? When do you think you can do a therapy for a disease? If you don't understand the underlying variation. So focus will be for implications for health and disease, the genetic variations that are seen in human beings, in healthy individuals and affected individuals. So to give you upfront, to get everybody on the same page, we have had the DNA double helical molecule, which was discovered several decades now. Okay, and I'm sure even the first year students among you would have heard of DNA, would have heard of the discovery, etc. So let us see over the last 50 to 60 years, what has been the progress as far as medical genomics is concerned. Where are we in talking about understanding diseases and treatment or cure of diseases? So the 1960s saw chromosomes and human disorders. Classical example of Down syndrome, where you have one whole chromosome extra, and that was the gross level at which one understood the human disease or chromosomal disorder or a syndrome. 1970s, we got the recombinant DNA in place. So that pushed from observational biology, from chromosomal biology, it pushed you to looking at molecular biology. You could look at molecular aspects using the restriction enzymes which were identified, which were put to use for doing recombinant DNA technology. So 70s and 80s saw flourishing recombinant DNA technology. Using these techniques, 1990, there was a very, very ambitious project, largely done in the West, where they said we will sequence the human genome. They thought that sequencing the human genome its entirety must actually get them all the answers to all the diseases. Genetic variations in health and disease therefore got a push, and 2000, that's literally 2 decades ago, there were a lot of efforts to understand and correlate genetic changes with health and disease. Now we talk about, in the last 10 years, everything is called as referred to as next generation sequencing or the next generation technologies, wherein you can sequence the whole exome, where you can sequence the whole genome, not just of humans, just about everything that you want. Okay? Somewhere along this, translation of these findings for clinical use, for treatment, for diagnosis, for cure was happening alongside. Since the topic is a little restricted to gene therapy, I'm just telling you the phase the gene therapy effort started in the 1990s. The minute they understood a little bit more of the genes and the genetic variations in different diseases, and then we have what is called as gene editing or genetic manipulation, actually at the level of the base change, which started in 2015. It's already 7 years, and you will hear a little bit more towards the end of my talk in these. So if you're with me on these landmarks in human genetics and medical genomics over the last 7 decades, I will tell you what we expected from all these efforts. 10 years ago, we thought there will be some DNA tests for different diseases. There will be sequences of several other genomes, not just humans. There will be gene functions which are understood. In 2020, that means already 2 years ago, we thought that there will be gene-based drugs for diabetes, hypertension, and all these common diseases. There was a hope that there would be very effective cancer therapy. There was also hope that you can go to the clinic and tell the doctor whatever your symptoms, and he would be able to look at your DNA profile and give you the exact medicine, the exact dose, which is called as pharmacogenetics or pre-prescription testing. Okay? They thought it will be a very standard practice for a lot of diseases. Then mental illness diagnosis, they thought could already be transformed because of the newer technologies. 2030, just a few years down to go, one expects there will be comprehensive genome-based healthcare. There will be computer models which will replace the human cells. Average lifespan will be 90 years. There will be major anti-technology movements. Let's say the genome editing, the CRISPR-Cas9 genome editing kind of possibility. And then for some of us who are like really serious about this area, there will be a lot of debate whether humans are taking their evolutionary path in their own hands. Having told you what we expected, I will now ask you how much have we achieved? Okay? So before I ask you these questions, I'll tell you how I have organized my talk. I will tell you a little bit about the human genome and the way we know it today. How do we identify a disease causal gene? How much of this is translatable for patient use or for societal use? When I say genetic findings translation, including gene therapy. And of course, there is another completely different aspect of complex disorders, hypertension, diabetes, cardiovascular diseases, and so on. How much progress have we made? Where are we? Or do we need alternate paradigms like combining Ayurveda with genomics? So this is going to be like 3 sections largely of my talk. And human genetic disorders. Now, if I introduce you, I already told you chromosomal disorders like Down syndrome, Klinefelter syndrome, Turner syndrome. These are simply because of one large chromosome, extra chromosome present or absent, or a large part which is deleted, and so on. Then another group are single gene disorders. You can speak for yourself there. Name a few common single gene disorders, which all the time you hear about, which you would have read from your class 10 onwards. Can you answer? Can you tell me a couple of names? Single gene disorders. Thalassemia, sickle cell anemia, cystic fibrosis, Duchenne muscular dystrophy. These are all caused by one gene, one defect in that gene, and it runs in families. That's about 6 to 8% again. Complex traits is the largest disease burden. I already mentioned to you, we are talking about hypertension, diabetes, cardiovascular diseases, schizophrenia, Parkinson's, so many of these things which you hear all around you. These form the common complex traits, 60% of them. Okay? But what is the situation there? Chromosomal disorders, you can here very clearly see here. I will try and use my pointer. Laser pointer I'm using. You can just see here, you have one extra chromosome 21, which makes it a Down syndrome child. Otherwise, all of us have 46 chromosomes. These kids will have 47 chromosomes. Klinefelter syndrome again, XXY. Turner syndrome, XO. So loss or adding of one will make a chromosomal disorder. But when you talk about single gene disorders, you are only looking at it at the genetic level, DNA level, or at the molecular level. You have to have families in which a particular disease is running. Parents may be unaffected, as shown here. They are only carriers, but there could be some children who are affected, some perfectly normal, and some, just like the parent, a carrier. But what is important here is it is just one gene, one mutation, which is going to be responsible. And therefore, when you're evaluating a population, everybody more or less will have the same mutation in that gene for a given disease, like say thalassemia or cystic fibrosis, because these are what you're familiar with. On the other hand, complex disorders are indeed contributed to by many, many genes. At this moment, we don't even know how many genes contribute to type 2 diabetes. 1 in 25 individuals in India suffer from type 2 diabetes, but we don't know the number of genes which contribute. Just to make you understand, I have only put 4 genes out here, and to tell you that along with genes, the non-genetic factors are also major contributors for disease onset. What does this mean? You can easily understand when you talk about a type 2 diabetes patient whom you have definitely seen around somewhere. They will tell you, they'll tell them, please don't eat sweets, go for long walks, do exercises, avoid obesity. These are all lifestyle disorders. And these are the non-genetic factors which actually contribute to disease development. So while A, B, C, D genes themselves can contribute differently in each individual, A and B may be a major one in one, B may be a major one, others are minor, A and C could be a major, and so on. You have non-genetic factors also which make it very, very susceptible to developing the disease. And this genetics or trying to get genetic basis of complex disorders have continued to remain a challenge in the last 30 years since the Human Genome Project. It continues to be 30, no, 50 years since we have it all in hand, and it continues to be a major challenge. And therefore, it's very important that lifestyle is taken care of. And ultimate goal in medicine is you want to have a diagnosis first for a disease, right? And when you talk about a diagnosis and a prevention, you're already at the stage to say, look, I need to understand the genetic basis. That means molecular diagnosis will be important for you to talk about prevention. If I have a gene, a mutation which is known for the disease, I definitely can use to go and screen families and prevent the disease. If you can't prevent, you know the old proverb, prevention is better than cure, right? So if your ultimate thing will be cure, but if you don't have a cure, you can only hope to prevent. If you can't have a cure, you at least hope to have a treatment. So this is the central dogma in medical biology. Diagnosis, prevention, cure, and treatment in that order. Okay? And overall, today we talk about P4 medicine. P4 stands for predictive, preventive, personalized, and participatory. So today's goal is this. All efforts in medical genetics is targeting P4 medicine. So when you talk about a gene therapy, it could be a treatment. If you talk about a CRISPR-Cas gene editing, you're talking about a cure. Okay? So that's the kind of a extent that we have gone to. So now I'll ask you, if we talk about all these, where are we in terms of understanding or achieving the goals of P4 medicine? I'll ask you a few questions. You can answer loud and clear. Your teachers will hear you and they will know whether you're right or wrong. Do we know all the genes in the genome? Do we know all the disease-causing genes? Do we have genetic testing for all diseases? Yes, no? Okay. Why do people respond differently to drugs? You come with a headache, somebody else has a headache, somebody has to take 2 tablets, and you are happy with just half a tablet. These are all differences which are there between individuals governed by genetic makeup. Do you have biomarkers for early disease detection? Can you have structures of proteins which can solve all the problems? I can go on. You can list. You yourself can ask 100 questions, okay, and see for how many of them you have answers. Okay. So we will see. Taking an example from each section, will tell you where we are. Then, before I go into the actual part, I will ask you, why should we not be using all the Western data? The West has advanced so much in medical genetics and genomics. Why can't we use that data? And should we really be working with human beings? Or can we not use animal models for all our work? Okay, in a lighter vein. Okay, so this is what the similarity between man and mouse is. All of you know that mouse, Drosophila, these are all very popular models to study genetics and diseases. Okay, now you can see, except for the X and the Y chromosome, which is here, which is conserved between humans and mice, for everything else, there is a lot of mix of different chromosomes in the mouse if I keep human as my reference. Okay? Another funny picture. This is how similar we are to our nearest ancestors, and yet we can't use them for all our experiments or for all our meaningful interpretations of translation, because there are basic genetic differences and there are limitations in some things. So we have to look at human beings and go ahead. If we talk about human beings, can we then not use the Western data? The answer is clear in this picture. This is just showing you with genetic markers, you can group populations. Okay? Black here, I hope you can see my marker moving around. This represents African population. The blue here represents Caucasian population. The green and the pink here represents Chinese and Japanese. And the green and the orange that you can see, the green represents North Indians and orange inside the green represents Gujaratis in the US. What does this tell you? That definitely there are population-specific differences. Although we talk about a large part of the genome being the same in all human beings, there are some population-specific differences. And therefore, utilizing different populations will be very useful. You are sitting in Kanchipuram, south of India. I'm sitting in Delhi. Okay? Won't you like to see the difference between North Indians and South Indians? if you see the same thing, African, Caucasian, Chinese, Japanese, I'm showing you. But if I make like what I showed you in this picture, North Indians and Gujaratis, if I separate out using markers, North Indian is green, South Indian is blue, but see here, they are still in one cluster. That means largely the Indian genetic makeup to a large extent is similar, but using specific markers, we can identify different populations. Okay, if there is a necessity to do those from disease point of view. Okay, so this is just to tell you that Caucasian, if I say, is red, Indian genome, I say, is blue, Chinese, I say, is green, African, I say, is black. Just see that there is an intermix of some bit of Caucasian in the Indians, more in the North Indians compared to South Indians, etc. This is just to get you a feel of population specific differences and why it is important to study different populations, what we refer to as trans-ethnic populations. Okay, so it's good to study these because a lot of meaning will come. With that background, I will now tell you very quickly about the effort which was made to get the human genome sequence. All of you are familiar with this? A double helical molecule of DNA is packaged as a chromosome which is present in the nucleus in all our cells which contribute the human beings. Always there is a diploid number in all the somatic cells. Only the X and the Y chromosome, that means the sperm and the eggs have 23, 23, which becomes 46 on fertilization. The whole idea of human genome sequencing was to look at these 23 pairs of chromosomes and try and get the sequence information. Remember, these are chromosomes, which is referred to as a karyotype, gross level, where you could have seen one extra 21, which made a patient Down syndrome. The sequencing to get the sequence information for all the 22 autosomes and the X and the Y chromosome. So what were the aims of the Human Genome Project? Sequencing the 3.2 billion base pair was a major aim. Annotation and understanding the hidden secrets there was the second one. Characterizing the genes and assigning them functions, they called it all as short term. So short term could be 5 years, 10 years, 20 years. And long term was supposed to be gene therapy, personalized medicine or pharmacogenomics I told you about. And of course, basic research will always continue. But are we there? When the Human Genome Project results were announced, this is a picture from an old age home. Everybody thought everything about all diseases will be understood, old age diseases will be conquered, and there was so much of happiness. But unfortunately, we are nowhere near understanding large number of old age diseases or complex diseases. What we have is an unexciting series of sequences, and you all know there are only 4 letters in the language of DNA, only 4 alphabets, G, A, T, C, permutations and combinations of which make the genes, make you what you are, and what we have is unexciting one. If I now gave you an exercise to understand what the sequences mean, it's exactly like taking a language textbook. Remove all the punctuation, spaces, capitals. Struggling? Some words you heard? Okay, I can hear you mumbling something. So some words you probably got, some words you can't get. And this is what we are doing with the human genome. We are trying to still make sense. Some words which are clear are something which we already know. New ones we are trying to understand. So if I gave you this paragraph, it's very easy. You can easily make out what is there in the previous paragraph, right? So this is where all our efforts are still going on to understand the secrets in the human genome. Substantial progress has been made as far as single gene disorders are concerned, but not for complex disorders. So just to tell you that these single nucleotide polymorphisms or the changes which make up the differences, why you don't look like your sister, or why you don't look like your brother or your father or your mother totally, is because there are multiple spelling mistakes in the genome, which are referred to as single nucleotide polymorphisms. Some of them can cause a harm, some of them may be having no effect at all. If I have to illustrate this with amino acid sequence as an example, if I said I'll take a car or a cab, it means the same thing, right? It's just that I'm using two different words. But if I said I will take a bar, bar is the word in the dictionary, but that's not what you started off with. Jar is a completely different word. All that I'm doing is single base changes or point mutations which are making these changes. If I translate that, because you're students of science and all of you know the amino acids coded for, TCA codes for serine, TCC also codes for serine because that's in the wobble position. Whereas ACA, if the T is changed to a A, it becomes threonine, and if T is changed to a C, it becomes a proline. Definitely, this is not the amino acid that you had in your original protein. Therefore, when you have a mutation, a protein can get changed, an amino acid can get changed in the protein position, and it can have a dysfunction or no function or a malfunction. Okay? I can also have spellings of different kinds. I can spell color as COLOR, or I can use the British COLOUR, or I can put characterize with a S or characterize with a E. These are all kinds of alternates and insertion deletions, etc., that I can have in the human genome. There are over 10 million SNPs in the human genome. That means there are many, many such changes. From my father's chromosome, I can have a G there. In my mother's chromosome, I can have an A there. And similarly, it can go on. So, series of them can make a haplotype, but each one is referred to as a single nucleotide polymorphism if it is commonly seen in individuals. But when you see such changes in an affected, in a diseased individual, then you call it as a mutation, okay? Because it makes a major change in the protein. Like in sickle cell, a glutamic acid is changed to a valine just because of one base change. And you should know what that base change is, right? And that makes the person suffer from sickle cell disease. So these are referred to then as mutations when it ends up with a phenotype. With that introduction, I will just summarize this part to say that many kinds of variations are there in the human genome. Only this little red portion, less than 2%, is what is coding for proteins. Everything else are non-protein coding genes, intronic, intergenic, and so on. And in them, you can have a lot of common variations which can be sitting, which can be 5 to 50% SNPs can be there, or you can have large chunks which can be duplicated, or there can be a very rare variant. Only one in a 10,000 individual will show that variant and so on. But what is important for us, if you're wanting to understand a single gene disorder, try to see what is the mutation, what is the protein defect, can there be something which can be done with gene therapy, and so on. We are focusing only on this less than 2% of the genome. The rest of the areas are really filled with what I told you, more than 10 million SNPs, which are there at different places in the genome. So instead of answering the questions after the Human Genome Project, what it opened up was a Pandora's box. You got lots of omics era activities. starting with genomics, functional genomics, pharmacogenomics, computational genomics, comparative, and on and on and on. Together with the DNA, you could also look at RNA, which is transcriptomics. You can look at the metabolite, which is metabolomics, or you can look at the proteins, which is proteomics. These are all products of what your genetic makeup is, what it codes for. Okay? So now let's see. So what I do in my lab is largely try to identify a gene which may be responsible for a particular disease. Let's say intellectual disability, commonly called as mental retardation. Okay, very common. One in 100 children are born with mental retardation. It's so common. Do we know all the genes? We don't. So we can definitely. So and I have other things, but I'm just telling you: intellectual disability, Parkinson's disease, schizophrenia. We don't know the genes for all of these conditions. We know some, we don't know all. So discovery genomics is one area. When you discover something, you definitely want to do functional analysis of that, which we use cell lines, or you can use animal models, or you can use embryonic stem cells, or what we today call as stem cell research. And we also look at some complex disorders, because all these are the ones which I told you have a definite mode of inheritance or Mendelian inheritance pattern. Complex trait, you could just have one individual, one family member in your large family. You may just have one person who has rheumatoid arthritis, only one person who has type 2 diabetes. Okay? They are not familial. They are just sporadic cases. But in a population, you will have several of them. So we'll also look at rheumatoid arthritis, ulcerative colitis, Crohn's disease, and so on in the lab. We try to develop new drug molecules. We try to see who is a responder to a drug and not a responder to a drug. Not just DNA, we also look at gut microbiome. Today, gut microbiome has become extremely important. What you eat is very important. You are what you eat. So when you talk about lifestyle, everything contributes. And then we combine genomics with Ayurveda, and that's another area. Why are we doing all this? Because we want to understand disease biology, and we want to understand or be able to predict disease and prevent disease. So that takes us to what we call as translational medicine. One way is with diagnostics and therapeutics. I mean, you can have diagnostics first, and then you can have therapeutics. Maybe there are not enough drugs. Novel therapeutics are very important. So this is broadly what I do. Now I'll tell you, when I said we don't know all the genes, don't worry about this. But those of you who have a book with your hand, please just write down this word OMIM. Google it later and see the details of this. Just to tell you that this gives you constant update on how many genes we know for a typical single gene disorder like Mendelian phenotype. Doctors say, hey, look, this disease is running in the family, but we don't understand the genetic basis. Okay? So we still have over 1,500 diseases for which we do not know the disease-causing gene. Okay? So you can imagine how much of work can be done alone in single gene disorders. So there is a lot of progress because of the new technology, especially the next generation sequencing. And now more and more disease causal genes are getting identified. I will just give you one example of why India is special, how you can be very good, you can be lucky, you can be very good researchers using Indian material and using all the technology which is currently available globally. Okay? India is very rich when I say with patients. Here is a family with intellectual disability. You can see all age groups. Older people, middle-aged, and you can see young people like this. So this is from one single family. If you read, if you have known how to look at a pedigree, This is a 4-generation family that we had. Only boys are affected with mental retardation. Obviously, that means it could be an X-linked character. If it is an X-linked character affecting only the boys, you know the mothers have got to be carriers. The gene or the mutation in the gene is always coming from the mother to the son. It can also go to the daughter, but because she has 2 X chromosomes, she is saved. But because boys have only 1 X chromosome, whoever gets that bad X chromosome from the mother out of the 2 X's, you end up with boys getting affected. This was supposedly thought to be as a fragile X syndrome, which is a common condition for mental retardation. But it turned out to be negative. And just to cut the whole story short, just to tell you, many tests were done on these, starting with chromosomes, looking at inborn errors of metabolism, looking at the fragile X mutation, looking at small and big changes in the chromosome, and finally looking at linkage analysis using markers on the X chromosome, etc. It turned out sure it was on the X chromosome. So using the next generation sequencing, we were able to identify that there was a gene on the X chromosome which is called as MID2, which was not reported before. 2014, we published this. And if you see OMIM, which I asked you to write down, and if you look for MID2, you will see our reference, because our gene was a discovery which got entered into OMIM. Like that, when you have medical doctors coming across very good families, if it is not a known disease or a known gene which is causing the disease, that is material for you to take forward for research. And if you are lucky, you can discover several disease causal genes. I'm not talking about general genes, I'm talking about disease causal genes, which we are still struggling to get. Another family, just to show, I won't tell you details, but just to tell you, consanguinity. These are cousins who got married. Naturally, because it is an autosomal recessive disease, you ended up out of 8 children, 3 children got Parkinson's disease, and look at the age at which it got. I said Parkinson's is generally an old age disease, right? But you can have familial forms of Parkinson, and here, because of one bad copy of the gene from the 2 parents to these 3 children, all of them got it in their teenage, they got Parkinson's disease. Okay? Again, we were very fortunate to get this family. The earlier family was from Chandigarh. This family was from Aligarh. Okay? From this family in Aligarh, we got this family. We could do next generation sequencing. We just looked at all the protein coding genes, that is exome sequencing, and we identified a gene in this and published it in 2016. Okay, similarly, we have many genes that we have identified for some of these diseases that we work in the lab. So when you talk about next generation sequencing, today it is the most popular and the best tool that you have to go and look for disease causal genes. This is the pipeline, and this is the workflow that you have to start with very good families where multiple members are affected. You look for the known genes, you look for whole exome sequencing, you do the data analysis, which is where your computational skills will come in, because you can use the pipeline that can be set up and you identify the variant, and then you go and see how many members in the family have that. You can use it for screening in the family members to see what is the pattern of transmission, whether they are likely to develop the disease, and so on. But if you have to do functional analysis to get a little bit more information, then it is not just one method. Today, we talk about an integrated approach for all functional analysis of a gene or a mutation, what it does. You have biochemical analysis, you have cell biology, you have computational biology, you have animal models, you have genetics, genomics people looking at additional stuff. So you have an integrated approach which will give you the function. Now, this is all very nice, hunky-dory, pip-pip, as we say when we talk about single gene disorders. But when we talk about complex disorders, where are we? We already, I told you that it is genetic and non-genetic factors. There are many genes which are involved. Thousands of studies have been done on almost all the complex traits, but all that we have is a very complex picture. Instead of one gene, one mutation, one phenotype, what we see, look at the color. These are 24 chromosomes that you can see as long ones. Each color is representing a disease group. Now, if I say there is one mutation is not showing up only for one disease. One mutation is showing up here for 3 diseases. One mutation is showing up for 6 diseases. So I'm not calling them as mutations, but I refer to them as single nucleotide polymorphisms, which contribute to multiple diseases through multiple shared pathways. So we are in a very poor situation as far as understanding complex traits are concerned. Disease risk prediction is next to impossible as of today. Lots of studies other than the American and the European studies, lots of other populations are also doing these studies. We have also done it. We have also identified some population-specific patterns, which we are now taking forward for drug molecules, etc. But those are only a minuscule proportion of success for complex disorders. So goals of P4 medicine in complex disorder is almost not there, okay? So I'm just going to introduce you and just leave it at that, just to tell you that we think combining Ayurveda, the conventional, traditional form of Indian medicine, with the genomic tools may be a very good way to get better answers. That is to tell you, if you went to a diabetes clinic, everybody with type 2 diabetes is treated as one. But in reality, we are a mixture of different Prakriti or constitution groups. Those of you who have at least heard of Ayurveda would have heard of Vata, Pitta, and the Kapha constitutions, broadly 3 categories. We are a mixture of them. So combinations can be total of 7, but primarily 3 combinations. And we think instead of taking everybody together, if we do genome analysis based on their Prakriti subgrouping, we can have much cleaner results. So we call it as Ayurgenomics, and we do some studies on that. Because in today's context, the need of the hour for P4 medicine is that you require to have robust biomarkers. You need to be able to predict disease. You need to be able to predict course of the disease, differential treatment, and you want newer therapeutics also, because the existing therapeutics are not sufficient. And that's why some people say, oh, I've been taking my medicine, but no effect at all, because each one's genetic makeup is different, and we don't have gene-based drugs or personalized drugs at the moment. So, but genetics alone will not address this. We need to have, to achieve the goal of P4 medicine, particularly in complex disorders, you need to have multiple omics with your Prakriti-based classification or deep phenotyping. You need to have genome. You need to have extensive clinical information, epigenome, proteome, metabolome, microbiome, so many data sets. You can't manually handle this. So you use what are called today as artificial intelligence and machine learning methods. And we think in a few years, we will have P4 medicine being at least partly in practice for complex traits. Now I switch gears from this point and tell you how much of this has actually come to translation. Okay, I already told you the differences. Chromosomal disorder can definitely be tested. If your clinician is aware, If the hospital you're going to is a good one, they already know when they assess the child or an adult with the disease, they know what diagnostic tests are currently available. So for single gene disorders, for chromosomal disorders, everything is there. For pre-prescription testing, I may have a card tomorrow in my pocket, which can tell the doctor what my constitution is, but we're not yet there. Newborn screening for inborn errors of metabolism. I will go into a little bit detail, something which is so doable and something which we don't do in our country. Then there is in vitro fertilization and pre-implantation genetic diagnosis, because people who are infertile want to anyway have a biological child. So in vitro fertilization, but you can use some of the genetic tools, diagnostic tools to prevent. What I'm not going to tell you is cancer diagnostics or actual details of stem cell therapy. And for complex states, I said there are no tests. It's only the doctor who says, oh, look, you have rheumatoid arthritis, I'll give you rheumatoid arthritis drugs. You have type 2 diabetes, I'll give you these drugs. So it is only for chromosomal and single gene disorder that you can have translation, which is very effective almost in all places in the country. Okay? So you can have different kinds of chromosomal rearrangements. It can be structural or it can be numerical. That can be done. Who generally gets tested? Individuals or families with illness, mentally challenged children. First thing you do is get their chromosome tested. The infertile couples or people with multiple abortions, because they can have a lot of chromosomal anomalies. And you can also, if you know there is a disease running in a family, you can do prenatal diagnosis. Okay? So if I talk about chromosomes, I already showed you chromosome picture, but today there are very powerful ways by which I can have my 24 chromosomes with 24 different colors. Okay, so any change, any extra chromosome, any translocation, anything can easily be identified because of the chromosome specific colors. If here, look at this picture down here. This is blue is background for all chromosomes. Suppose I want to look at 2 chromosomes which I suspect has some, it's important for a lymphoma. I know that this green chromosome got a part of this pink, and this pink part went, the green part went on the pink and the pink part came on the green. One each of the chromosomes are okay, one of them got translocated. If something like this happens, either it is a disease. If it happens in an infertile couple, you can be sure there'll be multiple abortions. Okay? You can do these kind of molecular cytogenetic analysis even in single cells. When you go to the in vitro fertilization clinic, the doctor says, okay, your embryos are ready, come back for implantation. They will tell a mother to be. Before doing the implantation, they can actually check for chromosomal anomalies, at least the common ones, 21, 13, 14, X, Y, which are all commonly involved in chromosomal syndromes. That you can do in the prenatal diagnosis stage. Either you can do in the first trimester. Earlier, it used to be only done in second trimester. Technologies have advanced. Now you can do in first trimester, both chromosomal testing as well as biochemical testing for known diseases. That brings me to something which I'm sure all of you will agree is something which is doable. Not be affected. Therefore, you can't say it's running in the family, but suppose you have a child, you can do the screening for these common conditions and prevent a whole lot of infant mortality. Okay? Generally, the onset is early on in the life, like maybe sometimes it manifests within a month, sometimes in 2 months, but very early on. And some of these are treatable. Others, you can identify the mutation, go back to the family. Next time the mother is expecting a baby, you can definitely screen for prenatal diagnosis can be done if they think that they don't want to have an affected child again. All that is required is when the baby is born, 24 to 48 hours after the baby is born, after the baby had its first feed and its metabolism starts working, you just prick the heel of the baby and take a drop of blood, which can be used for so many different tests of metabolite screening. This is really a classical example of genomic medicine: predictive, preventive, personalized, and participatory. Suppose a mother says, no, I don't want my child's heel to be pricked. It's a newborn. She is going to be the loser. But on the contrary, if that is done, if everything is fine, mother is happy. If by chance they say, no, your child seems to have a problem, then they can immediately start the treatment because it is a metabolic defect. We are not talking about a gene therapy there, but only talking about a drug therapy or a diet therapy. Please look up phenylketonuria. Those of you who don't know, go look up phenylketonuria, which is only a drug-based therapy which is required, which can prevent these people from having mental retardation. So, more than 60 years now, since till now, it's more than 60 years since newborn screening is routinely done in the Western countries. Okay, you don't have to wait for a person to fall terribly ill. You can already screen a newborn and help the newborn to have a healthy childhood. But in our country, we don't have anything. Newborn screening is not mandatory. So we were again lucky to be able to do a study in Delhi State with our clinical collaborators, Professor Seema Kapoor and 20 other clinicians from different hospitals in Delhi, and a private partner who could facilitate this large kind of a study because logistics is very difficult. So we were able to screen 200,000 newborn babies for 36 different diseases. Okay, and we could save 1 in 200 newborns. And of course, it is very, very cost effective then, because once you identify, you put the child on the treatment, the child grows up to be normal, which means an earning member and not a burden to the family. It's a classical example of translational medicine and a technology for the people. Okay, just a simple newborn screening for metabolic disorders. These are the kind of conditions that the children end up with if they're not treated in time, which means they're not diagnosed in time and they're not treated in time. This is what happens. This is a classical example of the first child who was suffering from congenital hypothyroidism and was not detected quickly enough, not put on treatment, and became a crippled, mentally retarded, dwarfed child. But because the family knew the defect, next time this girl and she is a normal child. I can't even get a picture like this from India because we don't really practice this at all. And this is something which all of you will be sad to hear. You know eunuchs, right? Hijras. You call them hijras, who are generally seen in the red light area, in the streets. They are begging, they are dancing, they are singing, because these are marginalized section of the society because we didn't do enough. They only have one mutation in one gene. When these babies are born, they have ambiguous genitalia and they will have a few other defects. The minute the doctor sees that there's ambiguous genitalia, or when we do one of these metabolite screening, you can immediately see that they are suffering from congenital adrenal hyperplasia, and you can start the medication right away. They may not be fertile, but they can be normal school-going and school-going children growing to adults. You will not even be able to make out that the person is actually a patient of CAH. If untreated, they are the ones whom you treat as hijras or eunuchs, what are called. Okay, so this is just 7 rupees tablet a day, lifelong. But that's all the treatment which is required to make them as normal people, and which is so preventable. And diagnosis is possible, prevention is possible, but we don't do enough. So there are many, many advantages of doing newborn screening. This has to be something which is done in every state in the country. We are trying to push. In Delhi, at least, they have started for common government hospitals. They have started for common conditions. But at least if that awareness is there, you can take it forward. Now I'll come. All this is possible. You can have tests, you can have diagnostic tests, you can have all kinds of interventions, but what you need to be careful today in the societal context is also the ethical, legal, and the social issues, which are called as LC in medical genomics. If you think, if they know that you have a mutation which is going to get you, say, for example, a breast cancer mutation, you have, an employer will say, hey, look, you're going to be a liability to me. You will be absent from work. You will ask for insurance or you will claim your medical bills. Insurance companies will charge a higher premium. Okay? So both in terms of employment, in terms of insurance, in terms of overall thing, there'll be discrimination. And our country is very famous for eliminating female fetuses because everybody wants a baby boy, right? And hopefully things are changing now. So fetal sexing is a punishable offense now. Every ultrasound clinic is being monitored that they don't give the information of the sex of the fetus, okay? But by doing genetic testing, you can easily do fetal sexing, which has to be again considered very, very carefully and prevented. Human rights. A mother knows that she's going to have a Down syndrome baby, but she still has the right to say, look, I'm willing to look after my baby. I will not go for a medical termination of pregnancy. Okay? So, if you do human genetics, if you do therapy, whatever kind, drug therapy or gene therapy, you should be very aware of ethical, legal, and social issues in medical genomics or in the practice of medical genomics. That brings me to my last part, where I'll introduce you to what you wanted me to talk on, gene therapy or stem cell therapy, whatever you want to call it. Everybody is reading today about designer babies. What does it mean? It's not that the natural way that you and I were born to our parents who were interested in making a family. They just, and whatever child was born, they were happy. Male, female, affected, not affected. That's what one lived with. Today one talks about wanting to have only male babies or females, or wanting to have an extra intelligent or extra beautiful or a genius and so on. So this is the whole concept of designer babies on one hand, which is for improving some traits. Others say, I want to design a baby because I don't want any disease in my child. Are we really in a situation to do that? For single genes, yes. For single genes, there are multiple other ways which I already explained to you. So do we need to do things which are not really at the moment ready for use? And one example, series of example is what I'm going to tell you. So the first part I already told you, the chromosomes, DNA, and the metabolite profiling based diagnostics, which is for single gene disorders, which can be easily done. Then you talk about gene therapy, where you're saying, look, I don't want to go through with medication. Can I already have a manipulated gene put inside me so that the protein, the right protein is always there? That is very wishful thinking, very good. But if I have to tell you upfront, in 30 years of gene therapy, I told you we started in 1990s, okay? It's close to 30 years. Except for 4 or 5 diseases, we don't have effective gene therapy because it's not that you can't. You can use recombinant DNA technology. You can put the right form of the gene in a plasmid and put it to the individual. But how do you put it to the individual? The vectors, what we call as the vectors for gene therapy, are still not there. The best ones are not there. You can have all kinds of difficulties. So it has been a very, very poor progress. Severe combined immunodeficiency syndrome, SCID, is one of the most effective gene therapy which has happened. Little bit in Duchenne muscular dystrophy, little bit in cystic fibrosis, and so on. But otherwise, gene therapy sadly did not take off the way it was expected. Otherwise, for all the 2,500, 3,000 single gene disorders, we should have already had gene therapy, which is possible. But the problem is with delivery of the correct form of the gene to the target tissue, okay, and to get it functional. Second part is not just putting a good form of the gene just by changing the right form of the gene into the thing. Here, you are taking your own cells and trying to make the change. I remember I told you single nucleotide polymorphism and mutation, point mutation. So you're trying to make one change. That mutation you want to correct and then put it back in your therapeutic mode. So somatic cells is possible. I will take an example and tell you. But when you talk about germline genome editing, where I told you, remember, beautiful daughter, beautiful, handsome son, intelligent son, these are all very wishful thinking, where you need to do germline genome editing, okay, but which we are not ready. I will go quickly with these examples. I'm watching my time. So I will very quickly tell you that when you talk about gene therapy, cystic fibrosis is one of the 3 or 4 or 5 very good examples where therapy has been possible. So you can use DNA, which is by way of you put the correct form of the gene through a vector. That's a therapeutic vector we talk about. There are all kinds, viral vectors and non-viral vectors. But success story has been rather limited. You can also put RNA. You can also put oligonucleotides, which will do the job of the protein that you're looking at and take it forward. And you can have a direct, so this is genetic material, that's therapeutic vectors. Then the next step is where you can have a direct delivery. If you use, say, something like adenovirus vector, you can use a nasal spray to put the correct form of the gene to a child who's suffering from cystic fibrosis. So you have a nasal spray through which this vector, which has the right form of the gene, goes in. You can have it through the nasal, which can go, and then, of course, it should ideally function not just at the lungs, but all tissues where there is secretion. Remember, CFTR gene is not just only affecting the lungs. It is the major target tissue, but you also have several other tissues where cystic fibrosis is affecting an individual. Or you may take these cells from an individual and do what is called today as your therapeutic intervention or gene, the CRISPR-Cas mediated editing, where you're trying to change the mutation of the gene, mutation in the gene of the patient, trying to grow it and then putting it back to the individual and hoping it will work. Because this is involving somatic cells, it is permissible. A lot of efforts are on for hemoglobinopathies, for thalassemias, for sickle cell anemia, for cystic fibrosis, etc. You have stem cell therapy or CRISPR-Cas mediated gene editing, getting the correct form in your own cells and putting it back to you, rather than taking some other gene and putting it back to an individual. So here it is the same genetic background where there is a lot of progress, and they talk about in vivo gene therapy, which is inserting, as I already told you, it can go and be there, or what I just told you, I can take out the cells of interest, I can add the transgene, I can modify it and then do it, or I can edit it and then do it. How come editing became so popular? Okay, it was something which was published long time ago, but Nobel Prize in Chemistry was given to Emmanuel Charpentier and Jennifer Doudna for the most important discovery, what they thought, for gene therapy or gene technology based gene editing, which is CRISPR-Cas9 based ones. Not going into the details, but they are referred to as genetic scissors because you can take your own cell, tell the scientist or the lab that, okay, I have this mutation, I want that to be corrected, and I want to have a treatment. I don't want to be living on drugs all the time, okay? So you can have multiple ways by which you can have your gene mutation corrected, so that ultimately there are multiple ways. CRISPR-Cas9 is one way. Before that, there were two other generations, which is called as talons and zinc fingers, which were also methods by which you could correct the mutation in the gene and put it back into the cell and make sure that there's now a normal protein. These are things which you can just read. Lots of reviews are there. But just to tell you that, yes, therapies are there. Somatic cell therapy is all fine. The problem comes when you're talking about doing selective selection of the embryos or manipulation of the embryos or sperm and germline cells, or rather, the spermatogonial and the oogonial cells. So, in other words, it's something like we all know eugenics and the practice of eugenics, which was there, okay, early times, where it was selective elimination of some races, selective elimination of the unfit individuals. That was being done by massacring. Here we are talking about doing genome editing and then eliminating all of those. But are we ready for it? Are we ready to really get this thing? We can now engineer the human race. You can take evolution into your own hands. And because of the serious ethical and social considerations, in the 2 years ago, they made this International Commission on the clinical use of human germline genome editing. Remember what I've told you. For somatic diseases, or rather for somatic cells, gene editing and correction of the mutation for therapeutic purposes is acceptable, because only that individual is going to be treated with that. Whatever happens is in that individual, with his consent, they're going ahead. But when you talk about germline genome editing, you're talking about the generation to come. So are you in a position to say, look, whatever changes are being made in my germline or in the embryo that you have, or you as a couple you have, do you really know whether the changes are really the right ones? What are the other consequences of changes which are not the desired changes, and so on? So broadly, these were the issues which had to be discussed, and some policy had to be put into place. So they said that a society, if at all they were willing to allow, today I told you fetal sexing is just not allowed in our country. Like that, tomorrow if they say, no way you can do germline genome editing, there is no way a laboratory or a doctor can do this. But if for some reason, tomorrow, if the policy says, no, for some things we will consider germline genome therapy, you need to have things in place. Okay, so we should take care of the societal and ethical issues, and we should see what are the broad implications. So, simply said, if I have to tell you, where are we? At the moment, no clinical use of germline genome editing is allowed, because further research is necessary, because we have no means as of today to say that there is only a desired change and off-target changes are not there. We have no way of saying or ensuring that. All this contradiction was started off because of this kind of work which was done by a Chinese scientist and which caught the attention of the world. He treated embryos to say that they will not be susceptible for HIV/AIDS, but which was not even correct, because there are multiple players which are involved in HIV/AIDS. But because they did not have the ethical issues addressed, they did not really have everything in place. The children were born, twins were born, but obviously it was not the right thing to do. So that created a furor the world over. And today we are not allowing clinical use of germline genome editing, but except in very, very special cases, they say when the time comes, when everything is available, when you have a chance to see that there are no off-target effects, maybe only two categories of individuals or families may be allowed in the first trial. Okay, I'll explain that in the last one, but not for categories like complex traits, not for categories which are applications which are not related to heritable diseases, like what I told you. You want a beautiful child or an intelligent child. None of those quantitative or traits can be actually troubled. And monogenic conditions that cause infertility, maybe, but those are all subsequent categories. Really speaking, if you have a couple who has a disease, both the genes are affected, but that is an autosomal dominant disease. Every child in the family, even though the mother is normal, every child is going to get affected, which means if a couple wants to have a biological baby, there is no way your gene therapy can help. Okay? Unless you do CRISPR-Cas9 gene editing. But if you think you're going to edit a gene, which is the mutation, do you know whether the technology is making only that change, or there are 100 other changes which happen in that cell because of the technological limitation, which is what the world is now trying to improve the technology. Or worse, I'm sure all of you would have come across families like this. Deaf mute people. A deaf mute is always marrying a deaf mute, generally. What happens? Most cases, they end up with all children who are deaf mute. Most cases, there are exceptions. In a situation like this, where there is no chance for a normal gene at all, or a normal gene is not there in both of the parents, if at all CRISPR-Cas9 gene editing is done, you may have a chance that you may have a child. So where all progeny are going to be affected is the only category which, if at all the technology is right, they may allow, but not other places where you have a chance of having a normal child or a carrier child and so on. Okay, so I close with that part to say that only for serious monogenic disorders, and if you have all the methodology in place, and where your parent is wanting to have, the couple is wanting to have a biological child, only then they should be allowed, but when it is ready. I want you all to read the last sentence here. You are building an expensive bridge to a remote island before we know if we are ready to use it. This was one of the several reactions to the report which was brought out on human heritable genome editing report. Okay, so in this journal, so this is so aptly put. Even if you didn't understand anything of what I said, if you understood the sentence, I read again. You are building an expensive bridge, okay, to reach an island before we know if we are ready to use it, right? So meaning to say, it is too premature to be using CRISPR-Cas mediated gene editing based therapy as of today for even the serious diseases. For somatic cell diseases, yes, you want to take a chance. The individual is suffering. He wants to take a chance. He can do it. With that, I'll close my talk. But all the work that I talked about from the lab cannot be done only by me. It's all because of wonderful clinical collaborators and genetics collaborators I've had with me over the years, and lots of students. And of course, funding, without which you can't do research. So my message for students is there is so much excitement in science. There is so much to be done. There are 1,000 questions which need to be, I'm only talking genetics. There are so many other branches of science. So there are so many questions which still need to be answered for in the interest of the humans, because all our work is anthropocentric. So I like to leave you with this slide, okay, which is what should be your motivation to go forward with whatever you take. And I wish at least a proportion of you will take up science as careers. Thank you very much.
Thank Thank you very much, ma'am. The session is now open for interaction. Can you please come forward?
Yes, hi. You have to hold your mic closer. I can't hear you very well. Please speak louder.
Can you hear me, ma'am?
Yes.
Ma'am, how can you prevent chromosomal abnormalities?
Sorry.
How can we prevent chromosomal abnormalities?
I can't make out. Can somebody repeat that to me?
Ma'am, how can we prevent chromosomal abnormalities?
Oh, okay, perfect. Sorry, I couldn't hear that, right? Okay. Chromosome. Very good. Very good to ask that question. A chromosomal anomaly resulting in an affected child can happen to any couple. In other words, the mother can carry a child with a chromosomal anomaly. Earlier, we thought Down syndrome, for example, was only in older mothers. But today, we know mother of any age can have a Down syndrome child. It can be a firstborn, it can be a secondborn, it can be a thirdborn. Okay? So, to prevent this, in today's context, a routine screening of pregnant mothers for the common chromosomal conditions is the best way. When I say common chromosomal conditions, Down syndrome, Klinefelter syndrome, Turner syndrome, and analysis of chromosomes 13, 14, and 15. These are 6 or 7 chromosomes which are very common in pregnancies. So if you go for, if a woman who's expecting a baby goes for a regular checkup, and if they do the routine screening, at least for Down syndrome, surely Down syndrome, which is otherwise 1 in 500 live births, can be prevented. If they suspect that there is ultrasound is routinely done, if they see that there is some kind of a physical anomaly, okay, which the ultrasound says, immediately the doctor, the gynecologist should actually be doing a prenatal diagnosis on this lady, take the sample, do chromosomal analysis, and establish if it is a chromosomal disorder. Because you don't need to do any molecular test. It is only standard karyotyping which is required to talk about a normal one. They do a G-banded karyotyping or high-resolution banded karyotyping to talk about a chromosomal disorder which may be there in the expecting mother, okay, in the fetus. So this is the general practice. And routinely, everybody is advised at least to go for a Down syndrome. Okay? And second is, when there is a family with multiple, a woman has multiple abortions, immediate suspicion is chromosomal disorder. So that's when both husband and wife should go and get tested for their chromosomal constitution or for their karyotype and have the medical genetics person look at it and have appropriate genetic counseling. I hope you got the answers.
Yes, thank you. One more question, ma'am.
Yes?
How do we find the genes responsible for this?
How?
How do we find the genes responsible for the diseases?
Yeah. You remember I showed you a big family with multiple boys affected?
Yes,
Do you remember? All of you would have seen a large family that I showed you, right?
yes,
So if I work out the minute a family says, look, there is this child with this anomaly. Again, the next child also is born with the same defect. the doctor should immediately make a family tree and work out the mode of inheritance. Is it a single gene disorder, or does it look like anything like a chromosomal disorder? Is it a dominant fashion? Is a parent affected and a child affected, or parents are normal, only children are affected? Which means you only look at the mode of inheritance. Mendelian inheritance will be established. Then the doctor will diagnose it as a particular condition. If the gene is already known for that disease or a mutation is known, you just have to go for molecular testing to an appropriate laboratory. If they say we don't know anything about this disease, but the big family is there, then the doctor passes it on to a genetics lab where they are doing the research, where they can use the appropriate tools and use the get people from the family, both affected and unaffected members, and work out, identify the mutation, and then use it for subsequent pregnancies or for screening the family. So you need to have a disease which is running in the family with a clear-cut mode of inheritance. And you should have an appropriate clinician look at it and identify whether it's a known disease or a new one for which no gene is known. And then the clinician should also be able to collaborate with researchers so that they can use the tools to identify the disease causes, variant of the disease causes gene. Family material is most important, in other words. Okay?
Thank you so much.
Yeah.
Government,
Yes?
the person has got any disease, genetic disease, can also affect the second part trainer.
One child, yes?
The first one child has got a genetic disease, and
Yes.
also affect the second one child.
Okay, so that
It's a genetic disease, so it's also affected on the parent.
So if I understood the question right, you're asking if the first child is affected with a genetic disease, can the second child also be affected with a genetic disease? Okay? Now, before you call it a genetic disease, you would like to go back and have the clinician tell you the name of the disease, whether inheritance pattern is known for the disease, okay? And if the child is getting labeled, let's say we'll take a simple example of thalassemia. Okay? So the child is diagnosed as thalassemia later. Okay? Everybody knows that it is a recessive disorder. So both the parents must be carriers. Immediately, they will do DNA-based testing for the parent and the first child. When the second baby is expected, they will do a prenatal diagnosis to see whether the baby has the good genes from the parents or the bad ones from the parents, or one good gene and one bad one. So if it is good genes both from the parents, the child will be normal. If it is one bad gene from each of them, the child will definitely get the disease. If it is only one bad gene, just like the parents. Okay, so that's the you have to go and identify, get the diagnosis. If there is a genetic cause known, you have to get the genetic basis established. Then you can do predictive. Then you have to be able to do prenatal diagnosis. Only then can you say whether the second child is going to be affected or not. But if it is mental retardation, which happened to the first child, because hospital neglected, there was oxygen, let's say it was a difficult labor, there was oxygen deficiency or there was oxygen deficit. For a minute, the child really did not get oxygen in the brain. You can have the child becoming mentally retarded. It has no genetic basis. Okay? But that is something which the doctor has to say. That because of these associated birth-related trauma, the mental retardation happens. Okay, and not necessarily the second child is going to be the same. But these are to be decided very, very carefully and critically by the clinician whom you are going to consult. Okay, but if it is a genetic disease, they should be able to say mental retardation is the most difficult, because you can never say whether the second child is going to be affected or not. Okay, you can't say whether the second child is affected or not. Okay, if it is a mental retardation because of one of these kind of causes. Okay.
Thank you very much, ma'am. That was really very informative. Next, I would like to call upon B. Journey of second in the series to end the session 7 with board.
Thank you very much. Oh, okay. Do I log off?
Just wait, ma'am. We have.
Okay.
Good afternoon, everyone. Good afternoon, everyone. I thank brothers and all to be part of the phenomenal event. I take this opportunity to thank our guest, Professor P.K. Thelma, who honored this event with her inspirational talk. We are very grateful for the time and effort you took to create this event. Today, we get an opportunity to learn about genetic disorders, complex disorders, and people medicines, genetic editing, and next generation sequencing, and more. This will surely benefit us for a lot of future income. Thank you so much, ma'am, for your precious time and making this event even more special with your presence. Thank you, ma'am.
I thank you all very much for being there, and I hope you were able to get part of at least what I tried to put across. Thank you very much.
Thank you so much.
Thank you, ma'am. Thank you, Ms. Jenny. Now it's time for lunch. I invite you all to have a lunch. The lunch is the J block. It rooms J1, J2, J3. Volunteers are requested to show the way. All the participants of workshop